好的，那现在欢迎各位回到我们研讨会的下一个阶段。
Hello, everyone. Welcome back to the next session of our symposium. I would now like to invite our second speaker. Our second speaker is Dr. Liu Qi. Dr. Liu Qi currently is a postdoc at Fudan University, where she studies the adaptive evolution mechanism of human complex trait. Now I will give the floor to Liu Qi, Dr. Liu. Thank you. Thank you for the introduction. I will first share my screen. I think it's about noon, so hello, everyone. Thank you, everyone, for organizing these events, and I'm very happy to share with you my current progress. I'm Liu Qi. I'm from the Human Phenome Institute. My advisor is Professor Xu Suhua, and this is actually my latest project on the mapping structural variations and of H. longitudinus and R. microphyllus reviews vector pathogen adaptation. First of all, just like to share with you the background. Well, in the evolution of human beings, pathogens actually has a great impact of building of humans and shaping of humans. Comparatively, we also shape the evolution of pathogens. One very classic example is about the Anopheles malaria. Anomalous mosquitoes would transmit these transmodiums into the human being, which would cause malaria. And where in areas where malarias are high, actually carriers of sickle cell defect genes actually have the high ability to resist malaria, which make it even more evolutionary. These kind of genes would then be preserved. Whereas in pathogens, what's the effect of pathogens on human being? We have this this triangle triangle map. So the way is that you can through maybe sucking blood with the sucking of blood through the bites, then it would transmit these microorganisms into body. I think you may be more familiar with mosquitoes. However, ticks is actually the second largest pathogens, second to only mosquitoes, and ticks are also the subject of my study. Ticks during his absorbing power would actually transmit a lot of pathogens and actually lead to a lot of different diseases, such as the viruses and spinaches, ratages. So in Taiwan, currently in China, the studies on ticks show that these different ticks and different types of ticks and can actually affect a lot of human beings, and there's variations, and it also affects different numbers of people, and it can affect a wide population, which shows that this is very risky and dangerous for human beings. And it is not only an influence on human beings, it would also have an influence on livestock production. But in order to prevent tick-borne diseases from happening, currently we don't have viruses that could actually prevent it from happening. We mostly control the production of ticks, but currently these chemicals, they not only pollutant to the environment, also say that it is also known that it would also create drug resistance within the ticks. So we need to find a safer and more environmental friendly ways, maybe through like a biological control. But there are actually a lot of studies shows that among different ticks, there are actually a variety of different types of ticks. This is actually the distribution of different ticks, and you can see that they are distributed in different areas. And this will make it difficult in different geological and different environments that would actually have an impact on the evolution of ticks. In 2020, a team actually looked at and first found a great variety of ticks and actually tried to find the genetic information inside. And it is found that with different ticks, they actually have some very classic geological differences. Also, for different ticks and also the pathogens distributions. After comparison, it is also found that the category of different ticks also have different characteristics. And since ticks sucks blood, through the transcriptions analysis, it found that the mechanism is also sucking blood is also different. So initially, Dr. Wei introduced that they through these introduction, they identified some very important genes and also some mechanism. My research also adopts a similar manner. We want to look at the SV and to see if SV actually has a difference and if that plays an important role. So the way I choose this is there are different types of ticks. I mainly choose two very iconic ticks. First is the H. longicrotis, the other is R. microplus. These two types, they represent different life cycles. In addition, H. longicronis is also most widely spread and affect a wider area. So that's why I select these two ticks. For each ticks, I used a sample of over 100, and the death is about 80%. So the death is actually not that high, I would say. Afterwards, I adapt 3 different tools to do an SV testing and analysis. And I try to analysis, injections, and combine a combination of SV and do some downstream analysis. First of all, let's have a look at this map. We actually created this map, and it shows, and we also look at in different morphologies, what does it look like? And in these 2 different types of ticks, you can see that in terms of its genomes, they're repetitive. They are quite repetitive in terms of the series, but the frequency is quite low. And in terms of H. longicrotis, the depletion is actually quite high, but in R. microplus, depletion, high frequency depletion is actually is comparatively lower. So this shows that in different ticks, the FV map is actually very different, and these may have to do with their genetic models and also have the choose of mechanisms. So that's why I look at the structure, the genetic structure. You can see that in terms of H. longicronis, they don't have a very clear clustering pattern. So samples from different geographical areas, they are actually quite similar. Only a few samples will be separated and different. However, in the R. microplus, they are actually have a very clear geographic clusters. So you can divide it actually into the central China, South China, and Southwest China. These results with the SMP model and also DNA model, I do the analysis and I find that this is consistent with the results that I previously done. And we also compare based on the differences, look at the among different groups, what's the SV number. And you would also know that in terms of L longicrotis, H. longicrotis, the number of SV is similar. However, in R. microplus, the SV differences are quite significant. And these differences is also quite significantly correlated. So this may seem that differences of latitudes may affect the genetic phylomorphism and local adaptation of R. microplus. The next step is that we also want to know in this SV what's its impact on the genomes. So we collected some very important genes that's important these this kind of species. So we find something related to sucking blood because they live on the blood. So this has to do with these different functions. So we had a look and we identify these SVs and to see on these important genes if they're actually a negative selection. And you find that only in we only find this depletion and repletion would actually significantly reduce in our microplus. So this means that these repetitive genomes and also the depletion genomes, it is actually undergone a negative selection in our microplus. And if you look at these numbers and also the overlapping situations on these genomes, for different biofunctions, you will all see these different SVs. But in these FCs, among all these groups, the frequency, I would say, is not high, or there's not a significant differences. So maybe in the future, we can continue to look into these FCs and how they affect the characteristics of blood sucking. Lastly, we want to have a look and see that in these different groups, if they're highly diverse and diverged, then what does that have to do with the development of these different groups? So we compared the different groups and also the SV diversion situation. And we identified that actually in CDSG, there is a 4 KB overlapping, so there's quite a divergence over there. So these segments is actually related to the C terminus of the CYPG gene. And through research, we find that the C terminus of the CYPG gene would actually prevent and prohibits these two genes from combining, and it would actually stop from binding the NZP functions and domains of TVB, which prevents it from simulating the NK signal pathways and affect the innate immune hemostasis. So we then use this information to confirm that once affected by the pathogens, the CPYG gene expression volume is significantly down regulated. So this means that the repetitive segments in the CYPJ may play an important role in H. longichronous in adapting and resistance to the pathogens infection. Now, since the one over here, the metaenzymas, is actually a common acronym, so maybe this identifying this gene would be important for us to achieve the role. At the same time, we also compare the R micro plus and also the different divergence, and have a look at this there. In terms of the proteinase D, we also identified 5 KB deletion, and this deletion is actually more significant in is completely deleted in the central and southeast China. So this is actually itself a gene that breaks down the red blood protein, and it shows that in different situations, then it would actually affect how it's expressed in the guts. And also the guts of the ticks is reported to be a very important organ to resist pathogens. So if we combine all these informations, then we come to the conclusion that in these deletions of these genes, it may have an impact on it would play an important role in the blood sucking habits and tolerance to pathogenic infections. So that may also be a way to control the ticks. Interestingly, we also compare the research in 2020, and we compare the distribution of these pathogens. And this is the characteristics that we identified in the Babesia pathogen. It is only occurred in the central China and the east-west China. And if we combine the information, and if we look at the distribution of the resistance of pathogens, this is what we've seen in the central and also southwest China. The host is actually the resistant cows and also infectious diseases. So there are hybrid species. So for this kind of ticks within its body, the SV number is actually quite high, and the distribution is actually quite high. We also see that the lesion of these segments is also it's also completely missing. But in the southern China, the characteristics are completely the opposite. So our estimation is that maybe the media and the host has actually have this co-evolution process. So in the future, probably what we will continue to do is that we will continue to use microplus as a vector, and also the distribution of pathogens and host. We will continue to conduct studies to look at the research. And for the H. longicornis, well, actually, there are two different types of reproduction methods. One is sexual reproduction, the other is parthenogenesis. So it's worth exploring the differences between these two different reproduction methods on the tick. And also, we want to understand more about the control of pathogens through the study of these to the ticks. Next, just to share that for the first time, we actually built the variation map of H. longitudinus and our microbes, and the genetic structure within the species was analyzed. We also understand the differences and the variation at the SV structure variations to immune response and highly differentiated among animals. And while identifying this variation, we're able to provide some insight for controlling these pathogens. So based on this experience, we actually developed a way to leverage this process. And lastly, I would like to thank my advisor, Professor Su Hua, for the support and for the guidance. I would also like to thank the Human Phenome Institute and my team, and also the international team, international project for supporting my project, and also like to thank SSE for organizing this symposium. Thank you, everyone.
谢谢刘博士带来的精彩的分享。我们来看一看，我们的参会人员有没有
Thank you.
问题。
Let's see if we have any questions from our audience members.
报告人肖博士提出了一个问题。
We have a question from our next speaker, Xiao Yun. So, if you think if there is a co-evolution relation between tick and the host, would it be beneficial or contribute to their co-speciation? Yeah, my question is that is it possible to facilitate the co-speciation between the tick and the host and to come up with a new species or a different species? So that would be associative speciation. Well, for past research, it's possible. At the moment, for the speciation or differentiation within the species, it would have a more larger impact within the species. But to come up with a new species, I do not have the answers at the moment. Or I have a question myself as well. I heard a report on how ticks are parasiting on the cows. For the secondary infection, the cows would be more immune to tick. And would that be something similar in human bodies? So that's the question on ticks, actually, on ticks biology. For humans as hosts, in my understanding, if a human body is infected by ticks for a second time, so the human's immune system would also be, so to speak, remembering the infection, and there will be new responses responding to the ticks. Okay, thank you. I have a question. May I ask? Okay, great. Dr. Liu, your research is valuable for us in terms of technology. My question is that the sequencing, resequencing of the, did you use long reads or other ways at the 80%? Short wrist. Short wrist at 80%? So the mapping results, what kind of mapping result can you achieve? For coverage, we can reach more than 90%, but the depth of sequencing would not be as deep. So, because at that time in our research, we considered it's not as had cover has wide in coverage. That's why when we selected the software, we decided to use the tools that are better at coverage. You mean just use the second generation of data to conduct coding, or did you call after reconstruction? And that was a direct calling. Yes. Sorry to interrupt, but due to time, let's pass the time to the next speaker. Okay, thank you. I'm so sorry, Susu. May I add on Liu Qi's? I want to add to what Dr. Wei was asking. If all the results are low, you don't have to call. You can use pair ends, and you can use split rates. For all the other indicators with reader depths, they may have different performances. Yes, because I used 4 approaches, so that covered all mainstream approaches. But for short riz, and our short riz results is around 60%. And that was criticized heavily when we submit the paper. And Dr. Wei, if you're interested, we're actually conducting a pen on all riz, and we did learn that short riz is not performing as well in difficult regions, but it was okay. It was doable in 3 or 4 years ago. Oh, we'll chat about this after the schedule. Thank you. Thank you for your discussion. Thank you, Dr. Liu, for your presentation.